I'm here at ASH in this cold Atlanta, snowy, and there's a lot of excitement in myeloma on various fronts, but very importantly on the various genomic aspects that we have discussed so far and will be discussing in the rest of the meeting. The main area revolves around understanding myeloma. One of the areas is how does a patient with smoldering myeloma progresses to myeloma and what genomic changes are connected with that. As we understand this, we can prevent progression of smoldering myeloma to myeloma. And I think that's one of the areas we are beginning to know what kind of mutations happen, what kind of genomic changes happen, and we are beginning to understand targets that we can go after. The second and very important area is that we know now that myeloma, as it progresses, it acquires new mutation. So we cannot just do one-time genomic analysis and be done. We need to repeat it subsequently. So when patient relapses, we'll do bone marrow and do it again because myeloma changes its color. And we need to change our treatment to match that or be ahead of that. And that's what is coming out very clearly in this meeting. The third thing is that we've identified certain mutations. And there are genes called RAS and RAF, FEM46 and others. And the significance of knowing this is that we have now drugs which can go after these genes. So we can now develop a personalized or precision medicine that can be utilized to treat patients. So for example, if patient has a RAF mutation, we have a drug called vemurafenib, and there's data being shown now that that drug can inhibit growth of these myeloma cells. Another exciting drug, for example, is patients who have this specific 11-14 translocation. They express BCL2, and venetoclax, which is one of the drug which goes after that target, is one of the most exciting drug in this meeting because that probably might be the next drug to be approved. So using these genomic methods, we can find what is specifically wrong in patients with myeloma and how their disease behaves, and we can treat the patient better doing that. And then finally, knowing these genomics, and we do expression profile, there are various ways of genomics. So when one says genomics, it's a very broad term. We look at the DNA and what are the mutation. We look at what is called transcriptome and look at RNA and see what genes have expression have gone up or down, and then various other epigenomic changes. So one of the important thing is that all these various genomic correlates are now being combined. So one of the presentation here by Dr. Shalat is that we are beginning to put everything together. It becomes three-dimensional, and then we can understand better and we can treat better. But the second or the final offshoot of this is that by doing this genomics, we are beginning to find new targets. So some of the new drugs which are going to come about now in next year or 2 or 3 might be based on what we are learning today, and that there are new targets in myeloma. We can use the drug to go after it and cure the disease or control the disease for a long time. So there are various very important findings that are coming up, which is going to change how we treat the disease, how we make patients better, and then how we understand to prevent it.